very good morning, my dear students. Welcome to this chemistry digital class. As you know that we are doing our chapter 14, that is biomolecule, in which we have already studied about the biomolecule and the four components of biomolecule: carbohydrate, protein, nucleic acid and lipids. These things we are studying. So in which carbohydrate we have finished, and today I am going to tell you about the protein. So let's discuss the protein with the help of these notes. Okay, so dear student, today's topic is amino acid and proteins, in which both are somewhere related to each other. How? Let's understand. Amino acid, first we will understand amino acid. These are the basic unit. If I will take it a little bit close, these are the basic units of protein, which contain two group, NH2 amino group and COOH carboxylic acid group. Okay, so if we will talk about the protein, then protein is the polymer of amino acid which is linked with peptide bond. These are the polymer of amino acid. It means that many amino acid, they will make a chain, they will form a chain, but how they will link with each other, how they will bond with each other, that is with the help of peptide bond. So you can say simply, these are the complex nitrogenous molecules which are essential for growth and maintenance of life. The protein is ensuring the growth and repairing of cell which is damaged due to some injury or due to some sickness. Okay, so dear student, if we will talk about amino acid and protein, so in this particular video I am going to tell you about the syllabus, the topics that is given in amino acid and protein in this particular period. So dear student, first let's understand about the amino acid. I have divided the complete class or complete period into two group, complete lecture into two group, that is amino acid and about protein. So if we will talk about amino acid, the first thing that we need to discuss is structure. So if you will focus here, the structure of amino acid is CH. There is 4 bond, 1 2 3 4. Fourth is H. So carbon can make 4 bond, in which one bond is made with hydrogen, one bond is the long chain of carbon, side chain, it can be many chain. One side it is carboxylic group and other side it is amino group, alpha amino group. So that's why it is called basic unit of protein, that contain amino and carboxylic group. Okay, so second we will study here preparation of amino acid, then nomenclature, classification and properties. So let's understand first preparation. It is very easy. The protein will, when hydrolyzed, that will hydrolyze into peptides, and peptides will hydrolyze further into amino acid. So it shows that the amino acid is the basic unit of protein. The amino acid is the basic unit of protein. Okay, like glucose is the basic unit of carbohydrate, amino acids are the basic units of protein. If we will talk about nomenclature, generally the name of amino acid is given by their tribal name. Tribal name is which depend upon the source from they obtained, from where their property is related. Okay, for example, if I'll take two example, glycine, sine. So dear student, in the end you will get the name ene. Okay, like carbohydrates you have seen saccharin. Okay, here you will see sine. So glycine word came from Greek, the glycose, which means sweet. So that's why its source is sweet, that's why name is glycine. Triosine, it came from triose, the meaning is cheese. Similar way, I will share this picture to you, the table of picture of this table to you, so that you can understand the natural amino acid, their name, their characteristics feature with side chain, their 3-letter symbol and their 1-letter code. So basically 1-letter code is the first word and 3-letter code is the first 3 word of the amino acid, that is glycine, gly, alanine, ala, valine, bal, leucine, leo, isoleucine, alo, arginine, arg. Okay, so these are the same way. So this picture I am sending, so go through this picture and you can easily understand what is the nomenclature of this. Okay, third is classification. So on the basis of number of amino and number of amino group and carboxylic group, the amino acid is classified into acidic amino acid, neutral amino acid and basic amino acid. So let's understand in detail. So first is acidic amino acid. These are the amino acid which contain more carboxylic group than amino group. mean carboxylic group is more than amino group, because dear student, one thing I just want to tell you in the structure. So when we are going to discuss the structure of amino group, suppose this is structure of amino gas, you can see here carboxylic group also is the amino group is also there. So here, due to lone pair, this hydrogen is going to be attached in NH2, and it will convert NH2 into NH3 positive charge, whereas after leaving the hydrogen, carbonyl group will become carbon negatively charged. So when we have negatively and positively charged, or I can say when we have acidic group and basic group, that kind of structure is called Zwitterion structure. Okay, that we will study in the basically properties. So amino acid is the one of the example of Zwitterion. So when it has acid and basic, if more acid is there than base, so obviously it is acidic amino acid. For example, aspartic acid, glutamic acid. This aspartic acid is also used in for diabetic aspartame. So glutamic acid. Neutral amino acid is when amino acid, those amino acids which contain equal number of amino and carboxylic group. So glycine, alanine, these are neutral. Basic amino acid, these are the amino acid which contain more number of amino group than carboxylic group. I think it is very clear. If more acid is there, more acid group is more than the basic group, then acidic. If it is equal, neutral. If basic is more than the acidic, then it is basic amino acid. Okay. With this, we are classifying the amino acid on the basis of its requirement also for our body. So there are essential amino acid, non-essential amino acid. If we will talk about essential and non-essential amino acid, so basically certain amino acid that can be made by our body itself, and therefore we do not require them in our diet. These amino acid are called non-essential amino acid. These are called non-essential amino acid because it is prepared by our body ourselves without any source. The human body can synthesize 10 out of 20 amino acid which is found in protein. Therefore, other must be supplied to our diet, and these are called essential amino acid. So out of 20, if 10 essential amino acid is prepared by our own body, those are called non-essential क्योंकि हमें उसकी ज़रूरत नहीं है बाहर से. But rest of the 10 amino acid which is not prepared by our body, that will require with the help of our diet. So that is called essential amino acid. These are valine, leucine, isoleucine, arginine, lysine, threonine, methionine, phenylalanine, and tryptophan histidine. Okay, these are the essential amino acid that is required by our body. Okay, so this is all about the classification. Now if we will talk about properties of amino acid, these are usually colorless, crystalline solid, soluble in water, high melting point, boiling point, contain both acidic and basic group. That's why it is called Zwitterion, and I have already explained how it is forming Zwitterion. When this H will attach to this NH2, will form NH3 positive ion and CO2 negative ion. This is acidic ion, this is basic ion. That's why it is called Zwitterion structure. This is all about amino acid. Now when amino acid will combine with the help of peptide linkage, then it is forming protein. So a little bit I'm going to tell you about peptide also. So basically, dear students, if we will understand peptide, the pH of hydrogen ion concentration on the solution at which the particular amino acid doesn't migrate under the influence of any electric field, that is called isoelectric point of that amino acid. Now peptides are compounds that is formed by condensation of two or more same or different alpha amino acid, जिसमें एक या एक से ज़्यादा अल्फा अमाइनो एसिड कंडेंस्ड हो जाते हैं, इकट्ठे हो जाते हैं, उस बॉन्ड को हम पेप्टाइड लिंकेज बोलते हैं. दैट इज़ द रिजल्टिंग बिटवीन कार्बोनाइल जो COOH ग्रुप होता है और जो NH2 होता है. अब NH2 we need to take one hydrogen from here, one oxygen from here. When we will hydrolyze, so it will convert CONH. Okay, why? Because one H and one OH, H and OH. H will come out from here and OH will release from here. So because of that, it will release water, and this is the peptide linkage. This is called peptide bond also. Okay, so for example, here I am going to show you. In case of glycine, if you will see, NH2 CH2 COOH plus NH2 CH CH3. This is alanine. When glycine and alanine will react, it will hydrolyze obviously H2. So one H from here, one H from here and OH from here. Okay, like this way we can see, as I told you, OH from here and one H from here. It will hydrolyze H2O, then we get this linkage, that is peptide linkage. If a large number of alpha amino acid 100 or 1000 are joined by peptide bond, the resulting polyamide is called polypeptide. Then we are using the word polypeptide and we are writing there N. Okay, so after bonding with the peptide, obviously we will get protein. It is a long chain of amino acid. So in proteins we are going to discuss about the classification of protein and structure of protein. Okay, if we will say the protein are the complex nitrogenous molecule which are essential for growth and maintenance of life. These perform a wide variety of biological function. Protein are the constituent of cell and therefore these are present in all living bodies. The molecular mass of protein are very high. Structurally, protein are long polymer of amino acid linked with peptide bond. So there are classification, structure and denaturation of protein. If we will talk about classification, it is most important in examination point of view. Classification on the basis of molecular shape, classification on the basis of structure and shape, classification on the basis of product form. So classification on the basis of molecular shape, it is of fibrous and globular protein. If we will talk about fibrous and globular protein, so it is there, like if we will discuss the fibrous protein and globular protein with the help of this book. So dear student, on the basis of molecular shape, first type of protein is fibrous protein. They have thread or fiber-like structure. They have thread or fiber-like structure in which polypeptide chain run parallel means ऐसे, and held together by hydrogen and disulfide bond. Held together by dihydrogen bond. These protein are insoluble in water, not dissolved in water. Keratin is the example which is found in hair, skin, nails, wool, silk, etc. And myosin, which is present in muscle. Second type of protein is globular. The word globular is representing spherical. So it is spherical in shape, in which chain of polypeptide coil around. Okay, like this alpha helix structure of protein. It is a globular because it is somewhere coiled. These are soluble in water, and insulin and albumin is the example. Second type of classification in which on the basis of structure and shape. There are primary structure, secondary structure, which alpha beta is there, which is most important, tertiary and quaternary structure. So if you will talk about primary structure, protein may have one or more polypeptide chain. प्रोटीन के पास एक से ज़्यादा पॉलीपेप्टाइड चेन हो सकते हैं. Each polypeptide chain has a large number of alpha amino acid which is linked to one another in specific sequence. The specific sequence in which various amino acids are linked with each other. Hope so you are understanding this line. It to form a polypeptide which is called primary structure. Okay, वो link जिसमें amino acid जुड़ता है, that polypeptide is called primary structure. So the change of just one amino acid in the sequence can alter its biological activity and can create a different protein. Second is secondary structure. Secondary structure are the conformation which the polypeptide chain assume as a result of hydrogen bonding. It is between carboxylic acid and NH group. The polypeptide is called secondary structure of protein. They exist in two different structures. So this is important. Now alpha helix structure, DNA alpha helix structure also you have studied. It is one of the most common structure in which polypeptide chain form all possible hydrogen bond by twisting into the right-handed helix. You can see here, if you will say CO double bond O, COOH is there, तो C double bond O, H2O, OH निकल जाएगा यहाँ से, तो H link कर रहा है. तो here you can see every H is linked with oxygen, every H is linked with oxygen, every H is linked with oxygen, and it is sure all the H hydrogen will link with C double bond O. And in this structure, the NH group of each amino acid unit is hydrogen bonded to the C double bond O of another amino, and it is present at an adjacent turn of the helix. Okay, so this is the structure. Dear students, once again take screenshot and practice. Nothing you have to do. You need to make a coil first. Then the coil, one carbonyl C double bond O will react with hydrogen. Hydrogen will link with N. So here you have to make inside the coil one coil you make carbon, and after leaving one you need to make N. Okay, similar with carbon, after leaving one coil you will write N. So carbon, nitrogen is on the coil, in between that oxygen and hydrogen is there. So when you will repeat this type of structure, you will understand this is the alpha plated structure. Now beta plated structure, all peptide chain are stretched मतलब शीट की तरह बनेगा near by maximum extension and laid side by side, which are held together internally with hydrogen bond. The structure formed resemble the plated fold. So here you can see here RCH, C is bonded with N, on the other hand side C is bonded with N, NCCN. So like this way, this is the structure. C double bond O, most important is this C double bond O, again bonded with hydrogen and nitrogen. N अब इस तरफ को है C double bond O bonded with hydrogen and N. So when you will repeat this type of pattern, एक तो आपको नीचे को बनाना पड़ेगा RCH, इस तरफ को नाइट्रोजन, दिस साइड का कार्बोनाइल, then this side again long chain of, mean you will this chain you have to make like this way. इसको आप zigzag बना रहे हो और zigzag को 2 zigzag 3 zigzag हो गए, तो how that one second and third chain will interrelated with each other, that is related with the hydrogen bond. This is beta plated. Now after this tertiary structure, the further folding of secondary structure of protein is known as tertiary. Further folding, अगर एक और fold आ गया तो that is called tertiary structure. It gives rise to the two major molecule shape, fibrous and globular. The main forces of stabilizing secondary and tertiary structure of protein, which is in which the hydrogen bond, disulfide bond and van der Waals forces and electron forces attraction are there. Quaternary structure, although many protein are folding, and but some of the protein are composed of 2 or more polypeptide chain, that is called subunit. The special arrangement of these subunit with respect to each other known as quaternary structure. So if you will understand in a very simple way, primary structure single line, it is like a bead of your chain, जो माला होती है उसकी तरह. Secondary structure is the fold in that bead. Okay, secondary structure is fold in that bead, one fold. Tertiary structure is one fold, then one more fold. You can see here this side parallel to this side is there. Okay, it means that here also somewhere they linked. Quaternary is manifold. The diagrammal representation of protein is the subunit of primary, secondary and tertiary. Okay, so if we will tell last topic, that is denaturation of protein. Protein found in biological system with unique 3-dimensional structure and biological activity is called natty protein. When a natty protein is subjected to physical change, like there is change in temperature, there is change in pH, the hydrogen bond are disturbed. Then as a result, globules unfold and helix get uncoiled. जैसे चेन की 2 साइड को निकाल देते हैं आप, जिप जैसे आप निकालते हैं, जिप पहले आप पहनेंगे तो जिप विल फोल्ड, व्हेन जिप यू विल टेक आउट तो बोथ साइड ऑफ जिप विल ओपन, इट विल ओपन लाइक दिस वे. एज अ रिजल्ट दिस इज नोन एज डीनेचुरेशन ऑफ प्रोटीन. डीनेचुरेशन, नेचुरेशन होगा जुड़ना, डीनेचुरेशन ब्रेक होना. So in which during denaturation secondary and tertiary structure are destroyed but primary structure remain intact because it is of single line. Coagulation of an egg white on boiling and curdling of milk caused due to the formation of lactic acid by the bacteria present in milk. These are the example of denaturation of protein. So basically denaturation is the process that changes physical and biological property of protein without affecting its chemical property. Okay, these are the basic denaturation. Acha, one left, that is classification. Third, on the basis of product form, that is simple, conjugated and derived. If you will tell about simple, conjugated and derived, it is on the basis of product form. So simple is what? These are the protein which give amino acid only by hydrolysis with acid or enzyme. For example, albumin, globulin, glutamines. These are conjugated protein. These are the protein which on hydrolyze give non-protein part or alpha amino acid. The non-protein part is called prosthetic group and it controls the biological function of protein also. Derived protein are the product and partial hydrolysis of simple and conjugated protein. For example, protons, polypeptides are there. So dear student, this is all about for today. Today's topic was amino acid and protein. So now I hope that you understood, although I will send the same notes related to the today's video, and you must write down in your notebook. You must maintain your notebook so that when you will come school from Monday, I will check your notebook side by side and you all must show your notebook with complete work. Okay, so finally the day came when we will going to be meet in school. But dear student, it's my humble request, by keeping all the precautionary measure, please, for your safety and your family's safety, you will join the school. So this is all about. So thank you very much. Have a nice day. Take care of yourself.